let me introduce the second speaker, Dr. Lijun Xu, Senior Scientist of Process Development Department of GenScript ProBio. Dr. Xu received her PhD degree in molecular bacteriology and infection from University of Copenhagen in 2022. After completing her PhD, she joined ProBio as a senior scientist in antibody process development department. She is responsible for external technical communication on cell line development and CLD platform and capability improvement. In this talk, Dr. Xu will discuss reaching IND faster with accelerated and high-yielding cell line development. Now, I'm delighted to hand over to today's speaker, Dr. Xu. Hello, Dr. Xu.
Sorry, I'm muted.
Okay.
Thank you very much for your really nice introduction.
You're welcome. Now you can share your screen.
Can you see my screen?
Yeah, you can play it. Okay.
Okay. So, yep. Hello and welcome. My name is Li Zhuan, and today we are talking about reaching IND faster with accelerated and high-yielding cell line development. First, an outline of the presentation. So I take you through the overview of ProBio's cell line development platform. Then I give you some detailed information about our host cells, TruK1 genes, and TruK1 ADCC+. Then I give you some detailed information about our optimized vector system, and finally a few words about our company ProBio. So first, the question, what is the cell line development? We define the cell line development as the process by which the cells act as living factories to produce therapeutic biologics, such as antibodies, vaccines, or other proteins of interest. It's a very early and crucial step in drug development. However, the significance of it might be overlooked sometimes. So selection of robust cell line can de-risk the project at very early stage and reduce the timeline, and also can save the resource and the cost. And a stable cell line can also guarantee the run-to-run comparability and deliver a more consistent drug substance. As a CDMO company, our primary objective of cell line development is to deliver a high-yielding and stable cell line in an aggressive period. So with this in mind, I'd like to introduce our ProCLD, ProBio Cell Line Development Platform. Here are the key features of our platform. First, we have two host cells, TruK1 genes and TruK1 ADCC+. The TruK1 genes is suspension adapted from TruK1, received from ECCC, and the ADCC plus cells derived from our TruK1 genes by knockout of Field8 gene to increase the ADCC activity. And then we have two proprietary and internally developed vector, DGV and UP. The DGV has been used for hundreds of projects, and the UP vector was recently optimized. And notably, we have shortened our timeline to 8 weeks from plasmid to top 6 clones, and the cell line deliver high productivity with maximum 10 plus gram plate titer, with the retention of high productivity after 6 cell doublings, indicating a very good stability. Some of these features that I'll be talking about throughout the presentation. The workflow of cell line development, actually it can vary significantly depending on many factors, such as different techniques, different host cells, and then different selection systems used, but it's always the speed limiting step. In our platform, the process is initiated with the codon optimization and plasmid construction, And from there, the optimized codon optimized gene is transfected or electroporated into our host cells by stable pore screening. In this stage, cell pores with high productivity are often selected in order to isolate the highly productive clone. With the optimized up vector, it takes less time to generate the stable cell pore than before. And we can also use the highly productive cell pore for quick production. Then we could get the samples as soon as possible to begin some pilot experiments. Following the pore screening, then we perform the single cell cloning, a very crucial and important step in generating a robust cell line. There are plenty of technologies that are available for isolating a single cell. Of course, the most commonly used today is still limited dilution because it's very simple and easy and cheap way, even though it's a bit inefficient. We also have the VIPs with the high seeding efficiency. It can also verify the monoclonality in a more reliable and straightforward way with the imaging capture. Then the single-cell clone screened with high titer and verified monoclonality will be evaluated in fed batch culture, and the titer and some potential quality attributes are compared to rank the top 6 candidate. And as mentioned, we have shortened the timeline to 8 weeks from plasmid, from transfection to top 6 clones, and it takes 4 weeks to rank the top 1 clone in AMBER-15. And the cell line stability is a key consideration when selecting the top 1 clone. So far, with the established cell line development platform, we have delivered more than 180 cell line projects, including different kinds of molecules. Around half of our projects delivered a monoclonal antibody, and 1/4 we delivered are different formats of bispecific antibody or trispecific antibody. And the rest are recombined proteins and some therapeutic proteins. All this indicating that it's a well-established and widely used and suitable platform for different molecules production. Recently, we have summarized our data, and the highest titer of MAP has reached 10 gram per liter. And as you can see here, around half of the MAP projects we delivered have a titer of 5 to 7 gram per liter. Only a few projects have a titer between 2 and 4. For the bispecific antibody, the average titer is 3.5 with the highest titer of 6.3. And even though the average titer of protein is only 3, but considering the multiple structures of different proteins, so the titer varies a lot with the highest titer of 13.6 gram per liter. We know that for every industry manufacturing process, to generate the product, we must follow the strict criteria to ensure the reproducibility, compliance, and also the quality. And for the biotherapeutic production process, there are some expected and minor deviation from replicates. So to assess the cell line stability, the cells will go through repeated passages, and then the cell productivity is evaluated in fed batch culture. In our platform, as mentioned, we guarantee the cell line stability with the retention of 70% of productivity up to 6 cell doublings. And the product should also have no clinical manifold differences. So that could definitely support the large-scale manufacturing. And so far, we have delivered more than 100 GMP batches. And here is some of the cases. So we show that the cell line maintains high viability over 80% through day 13, and the cell performance is comparable among the 3-liter production during process development and different 200-liter tox batches, and there is no significant variability throughout the scaling up process. Regarding to the titer and also the quality, the titer we achieved is about 5 grams per liter with very good stability, and the CEX profile also indicates the comparability among different batches. Then let's jump into our host cells. Actually, we commenced our mammalian cell line development service quite early in 2008 with the host cells DG44. We know that DG44 requires several rounds of selection with increasing concentration of MTX, resulting in multiple copy inserts, maybe up to thousands of gene copy. So it takes extremely longer time to get a stable cell line. So in 2015, we dropped the DG44 and shifted to TruQ1 SP, that is suspension adapted from TruQ1 received from ATCC. It's a glutamine synthetase-based selection system and only required single round of screening, thereby incredibly reduced the selection time. So using this system, we achieved a titer of 3 grams per liter. In combination with the new host cells, we also developed the GGV vector that has been applied to the cell line development projects. The until 2018, 10 years after we first initiate our mammalian cell line development service, we obtained the license of TruQ1 from ECACC for commercial use and sublicense agreement. And last year, our optimized up vector has been applied to commercial use. Here we show that the development of host cells TruQ1 genes. So it was initiated with TruQ1 cells received from ECACC and further adapted to chemically defined media. And when the cell fully adapted to serum media with short doubling time, it was assessed for productivity, stability, and scaling up. And later PCB and its derivative MCB were generated, followed by characterization respectively. For the characterization, we confirmed that there were no bacteria, fungi, or mycobacteria in the cells, and there were no mycoplasma and virus contamination. So finally resulting in TruQ1 genes. And by the way, you could refer to this DMF number to support your IND filing. In recent years, there is a consensus that non-fucosylated IgGs possess dramatically enhanced ADCC activity due to the improvement of receptor binding capacity without compromising of the CDC or antigen binding ability. There is a variety of ways to reduce FC called fucosylation. In our platform, the fucosylation activity was completely removed by knockout of FUT8 genes. This gene catalyzes the transfer of fucose from the GDP fucose to unlinked glycopeptide. So the construction of ADCC plus cells is initiated by transfection of TruQ1 genes by FUT8 disruptive plasmid followed by screening, and later the single-cell clone was performed to isolate the positive candidates, and the Sanger sequencing was used to confirm the true knockout of the target locus. And later PCB and its derivative MCB were generated, resulting in our TruQ1 ADCC plus cells. And of course, we have done the repeated passages over 6 cell doublings to make sure the knockout can be stably passed on to the production cell line. Here is a case study to show the non-fucosylated IgG produced in ADCC plus cells gained increased ADCC activity. As you can see here, the affinity of the Fc to the receptor CD16A is increased a bit. And when we both use the traditional PBMC-based method and Lucifer report gene-based method, we can see that there is an increased ADCC activity compared to the IgG produced in the Chuki1 cells. And as mentioned, we also assess the cell line stability by repeated passages followed by fed batch culture. So notably, there's only 4% decrease in productivity after 65 cell doublings, indicating the cell line is quite stable, and there is no significant difference in impurity produced in both the Chuki1 genes and Chuki1 ADCC cells. And the main peak displayed on the SEC, HPLC, and non-reducing CESDS profile are above the 95%. It's an average purity we achieved for the monoclonal antibody. And the N-glycan profile also showed that the activity produced by this cell completely lack focus in the glycan structure after repeated passages for 6-5 doublings. Then more details about our vector. As described in previous slides, the UP vector has been applied to commercial use since last summer, and we modified and optimized the original vector by adding tighter UP elements, resulting in UP vector. So compared with the original vector, it can insert into the transcriptional active region of the chromosome with high integration efficiency, and it can also shorten the timeline in the stable pore stage, as I mentioned. And notably, it can significantly improve the titer of difficult to express molecules despite its structure. So based on our experience, it's suitable for expression of different molecules of interest, including monoclonal antibody, bispecific antibody, and therapeutic proteins. So following, I'll show you some case studies to implicate the properties of UP vector. So in the first case, UP vector was utilized to produce a monoclonal antibody. So as you can see here, compared to the reference, the proportional high titer cell pores has been significantly increased, which can greatly improve the screening efficiency. And accordingly, the higher titer cell pores have been significantly increased, was dramatically increased, and it's reached 3 grams per liter. The productivity of cell pore and clone were also comparable. It means that the cell pores are relatively homogeneous and can be representative. The cell pores can be utilized for early process development to reduce the timeline. And the purity of the product expressed by both the reference and the UP vector was highly comparable, or above 98%, showed in the SEC HPLC profile. In the second case, UP vector was used to produce a protein. It's a therapeutic protein that is really hard to express by the reference vector. So the cell pore titer first was only 0.05 when using the reference vector. And compared to the reference, we can see that the UP vector increased the titer significantly to 0.5. It's, yeah, yeah. Sorry, I couldn't hear you clearly.
Yeah, could you please kindly share your slides again?
Okay. Yeah, sure.
Yeah, thank you.
Can you see the screen?
Not yet.
Okay. Just wait a few.
Yes. Okay. Now,
Sorry, I couldn't hear you. Is that working?
Go ahead.
Okay. Yeah, just go back to this case. So the up vector was used to produce a protein. It's really hard to express. So first, the cell pore titer was only 0.05 when used the reference vector, and then compared to the reference, the average titer of the cell pore was above 0.5. It increased significantly, and the final titer of the top clone reached 1.2 gram per liter. We also assessed the cell line stability by repeated passages and then evaluated in fed batch cultures. And we have optimized the fed batch process by modulating the feeding strategies and the process parameters, so the titer is further improved to 3 gram per liter. Anyway, after 70 cell doublings, the productivity retained 80%, suggesting the cell line is quite stable. Well, in the last case, again, a therapeutic protein was expressed using up vector. It can definitely boost the production because it was with a really high titer of 13.3 gram per liter. So far, it's the highest titer we achieved in our platform. And the protein expressed in top 6 clones are all displayed high titer and high purity as suggested by the SEC HPLC results. The mean peak reached 95%. All these case studies indicates the up vector as a powerful tool to manufacture your product. And recently, we have also optimized our vector strategies, especially used for the bispecific and multispecific antibody production. Here I listed some of the formats of the bispecific antibody we delivered. So we delivered the normal bispecific antibodies, the asymmetric bispecific antibody composed of 2 or 3 different polypeptide chains with or without the knob-into-hole design, and also the cytokines or nanobodies fused into the N or C domain of monoclonal antibody. And in addition, we are also experienced in assembly in vitro. For this one, the 2 different scFs were separately expressed in TRPV1 genes and purified and then conjugated in vitro to form a bispecific antibody. And we also deliver a tri-specific antibody that already obtained the IND approval in both China and US. For the bispecific antibody, we know that one of the most challenges is the random assembly of distinctive heavy and light chains because it will lead to the non-functional or monospecific impurities. So in ProBio, we first used the single vector system. We put all different polypeptide chains into one single plasmid. And then we shifted to the optimized 2-vector system. The different polypeptide chains are expressed in separate plasmid. And then we can modulate the relative expression of different chains according to the chain-to-chain ratio. And in the pilot transfection, we test several chain-to-chain ratios. Then 48 hours after the transfection, we run SDS page to get the first sign of the assembly. So it allows us to select the optimal ratio that turn in the productivity of correct assembly to continue the project. Here is a real case to show that the significantly increased productivity and purity achieved by this optimized two-vector system. So the bispecific is composed of two different polypeptide chains. It's asymmetric. And when we first expressed this bispecific antibody in one single plasmid, there are three strong bands as indicated in the SDS page profile. The top band is right assembly of two different polypeptide chains. Below are undesired homodimer, and then the single polypeptide byproduct. So obviously, this chain is overexpressed, leading to these two impurities. So accordingly, several peaks were displayed on the SEC-HPLC and non-reducing CSDS profile. So we shifted to the two-vector system. We put the different polypeptide chains in separate plasmids and tested several chain-to-chain ratios and found out that the best ratio is 2 versus 1. As you can see here, there is only one strong band in the SDS page profile, suggesting the most polypeptides get into right assembly, and the main peak in the SEC-HPLC and non-reducing CSDS increased above 70%, suggesting the significantly improved purity. And the title of the cell pool fed batch samples also goes from 0.5 to 5.4, suggesting the significantly increased productivity achieved by the system. Then finally, a few words about our company. Jessica has given a bit just now, so I skip this one. And then we are a global CDMO company with broad footprint and customer base spanning from China to all over the world. Our global headquarters is located in Nanjing, China, and now we have additional manufacturing site in Zhenjiang, and now we are in the process of extending our global manufacturing footprint to US. So far, we have established a fast-growing global track record. With the platform, we have delivered more than 180 projects for R&D filing. Most half of them are CMC or CMO projects, and the most advanced project delivered has already obtained the EUA from NMPA, and 23 of them has obtained the global R&D approvals, and we also have more than 100 GMP batches delivered. Okay, I guess that's all I have for the presentation, and any questions are welcome.
Thanks for Dr. Xu's excellent presentation. If you have any questions, please type it on the chat or Q&A panel. As you enter the questions, please also follow our LinkedIn account, GenScript Bio. There will be more online sharing resources, and we will share them on our LinkedIn at the first time. Thank you. Yeah, okay. We received some questions from the audience. Let's take a look at the first question. Dr. Xu, could you please answer the single vector system or two vector system? Which one do you prefer to use for a monoclonal antibody?
Well, normally we use one vector system to produce a monoclonal antibody. It means we put both the genes expressing light chain or heavy chain in one single plasmid. It's because compared to most bispecific antibodies, the structure of the map is relatively simple. So we can do this to achieve really good results. And another reason is that we have two CMV promoters in the one single plasmid, and you can put the different chain under the control of two different CMVs. Then we can get a actually the light chain will express more than heavy chains. Yes. So we don't need to use a two-vector system to produce a monoclonal antibody.
Okay, thank you. And the next question is, what is the advantage of the stable cell line for antibody production in comparison to transient expression?
As indicated by the name, it's a transient production. So the system is only a temporary solution. It's because the plasmid cannot be replicated in mammalian cells, and after repeated cell doublings, the plasmid will be diluted and eventually lose the productivity. But for the stable cell line, the transgenes will be inserted into the chromosome and copy with the genome. So it's quite stable and can achieve optimal productivity. So it can also guarantee the batch-to-batch comparability. And it also easy for the cell and protocol transferase. Yes.
Okay, thank you. Here is another question. What will I receive upon project completion?
Of course, you could get the cell line we generated. It's normally several vials. With each vial will contain the 10 million cells, and also we will provide you a fully recorded report, including the experiment design and the results and conclusion, and also some reports such as plasmid construction and transgene sequencing, and also COA. And yes.
Okay, thank you. And the next question is, do you have WGS data of the TROK1 gene as cells?
Actually, we didn't sequence the TROK1 genes. It's because the true cells is quite fickle. It means that it gets a mutation even under normal passages. So actually, we couldn't see any reason to do the sequencing of the TROK1 genes after suspension adapted to our cells.
Okay, thank you. That's all the questions we have received by now. Thank you for your questions and thank you for Dr. Xu's answers. If you have any more questions, please kindly contact us through our official email. Let me join others in thanking Dr. Chen and Dr. Xu for their time today. Thank you for attending today's webinar. Look forward to meeting you again soon. And we will hold another webinar about integrated one-stop solution for mRNA CMC. Please follow our linking. Yeah, okay, I got another question from the chat panel. Any experience on control? Okay. Oh,
Yeah,
also,
yes. Yes, I could see the question. Any experience on controlling the ratio for different chains or bispecific in building a stable cell line? Actually, the case I showed in the slides, it's an example. Of course, we have, of course, for the trispecific antibody, it can also indicate that if you control the ratio for different chains, you can get a relatively good results. So the answer is yes. We have a lot of experience on it. Yeah.
Yeah, okay. If you are interested in our service, please kindly contact us through our official email. Yeah, thank you for your time today, and keep follow our LinkedIn account, and we will share the latest news and our recent events on our LinkedIn. Thank you. Thank Dr. Chen and Dr. Xu. Thank you.
Thank you. Bye. See you
Thank
around.
you. Bye bye.